Okay. All right, everyone. Thanks for coming today. So my name is Kelsey Nasser. I'm a sixth-year cancer biology student. And I am more than happy to welcome Dr. Felicity Newell here. And it's kind of cool that we're able to do Zoom because she's all the way in Australia. It's 5 a.m. her time. So thank you, Dr. Newell, for getting up early. So just a little background, Dr. Newell is a senior research officer within the medical genomics group at QIMR Berghofer, I hope I said that correctly, Medical Research Institute. And Dr. Newell completed her PhD at the University of Queensland and also a Master of Information Technology at the Queensland University of Technology. And since then, she's worked as a bioinformatics programmer, developing software for the analysis of cancer sequencing data. And she did her postdoctoral research at University of Queensland and Queensland University of Technology using bioinformatic approaches to study cancer and autoimmune diseases. And currently, as we'll hear today, her research involves using next generation sequencing data to understand the genomics of melanoma. And this is part of the Australian Melanoma Research Project. So we're really excited to hear this. And it's really cool that her work also includes rare subtypes of melanoma that sometimes are not discussed as frequently. And I just wanted to do a quick reminder, any students and postdocs that would like to do a meet and greet with Dr. Newell, please stay on the Zoom call after the talk and we will do the meet and greet then. And with that, I'll hand it over to Dr. Newell. Thank you.
Okay, I'm sharing the screen now. Please let me know if you can't see it. So thank you for the invitation to speak today. So I will be speaking today about the genomics of melanoma subtypes based on our current project where we're looking at the whole genome sequencing of 570 tumors. So just starting with the CME Learning Objectives, at the conclusion of the presentation, the participants should be able to differentiate between the genomic characteristics of melanoma subtypes, describe the prevalence of point mutations and large structural rearrangements in melanoma subtypes, discuss the extent to which ultraviolet radiation-induced mutations can occur at different melanoma subtypes and at different body sites, and summarize the gene aberrations that commonly occur in melanoma subtypes. So just start with a brief background. So melanoma, which is the malignant tumor of pigment-producing melanocytes, has a high incidence in various populations in the world. So this can be seen in this graph here, showing the incidence in blue in males and females in various populations. And so it has a very high incidence in North America, is a top 10 cancer in most European populations, and the highest incidence in the world is in Australia and New Zealand. So this is a plot of the rates of melanoma diagnosis in Australia, with the red being higher rates of diagnosis. So I'm based in Brisbane in the state of Queensland, and we unfortunately have one of the highest rates of melanoma diagnosis in Australia. So when we're referring to these high incidence rates, we are referring to cutaneous melanoma affecting the skin. However, there are rarer subtypes which can affect other body sites. And these include acral melanomas. So acral melanomas occur on the non-hair-bearing skin of the palms of the hands, the soles of the feet, and the nail beds of the hands and feet, which are known as sublingual sites. So acral melanoma is relatively rare in European-derived populations, but is the most common form in a number of Asian and African populations. So mucosal melanoma can occur on any internal mucosal membrane surface, including the upper body sites, such as the oral and nasal cavities, and lower body sites, such as the anorectal and genital tracts. It is a rare form of melanoma with a poor prognosis, and this poor prognosis is usually due to the fact that it's diagnosed late due to a lack of early signs and lesion location. Uveal melanoma is the most common, occurs within the eye and is the most common intraocular tumor in adults. About 50% of patients go on to progress to metastatic disease, where there are limited therapeutic options. So in the last 10 years, there's been intense study of the genomics of melanoma, and these are primarily focused on cutaneous melanoma, though the rarer subtypes are becoming more studied in the last few years. So there have, of course, been many studies. I'll just mention one here, which is the TCGA, the Cancer Genome Atlas, pan-cancer analysis of various cancer types. And they published a paper in 2015 in Cell looking at the genomic classification of cutaneous melanoma. So this was looking at the whole exome sequencing of 380 cutaneous melanomas. And I'll come back to the results from this paper throughout the talk. So I'm part of the Australian Melanoma Genome Project. So that's a collaboration between my institution, which is QIMR Berghofer Medical Research Institute in Brisbane and Queensland, and a number of institutions in New South Wales, in Sydney, including the Melanoma Institute of Australia. So the aim of this project was to perform whole genome sequencing of more than 500 melanoma tumours, with the aim to perform various studies, including studies of the genomics of the rarer subtypes of melanoma. So the work I'll be talking about today is from towards the end of this project and looking at the whole genome sequencing of 570 melanoma tumours. So we also sequenced the match normal for each patient so we can identify both germline and somatic mutations within the tumour. And as we're doing whole genome sequencing, we can call the whole range of variants from the small coding mutations, which are shown in this circos plot here, with the coding mutations listed in the text around the outside. We can also look at copy number gains and deletions shown here in the red and green, as well as structural variations, which are shown here in the lines. So just a quick overview of the cohort. So as I mentioned, we have 570 whole genome tumours, and the work we'll be talking about has been published in a few papers in recent years, and also we are doing a larger comparison with a higher number of cutaneous melanomas to compare the different subtypes. So the first paper was primarily focused on cutaneous melanomas. We also, in the last couple of years, have published the rarer subtype specific papers for acral, mucosal, and uveal. So most of some 230 of our tumours are primary tumours, and the rest are local or recurrence or metastasis tumours. So the majority of as we're an Australian-based project, the majority of our tumours are from Australia, and therefore most of the patients are of European-derived populations. We also have a number of samples from our collaborators in China, and these are mucosal melanoma samples. And we also have some other tumours from collaborators in the United States, Switzerland, and Sweden. We also have matching RNA sequencing for about 180 of these samples, and 144 that have methylation data in the form of Illumina EPIC methylation arrays. So just mentioning the primary sites of these the various subtypes. For the cutaneous, we have these from fairly equal numbers from various sites from the body, from the ones that are more sun exposed, such as the head and neck, to those which were in general less sun exposed, such as the trunk. For acral melanoma, the majority are from the sole of the foot, and this reflects the general incidence rate. Most acral melanomas do occur on the foot, but we also have some from the palm of the hand and subungual sites, which is the fingernails and toenails. For mucosals, we have about equal numbers of tumours from the upper body sites, mostly oral and nasal, and from the lower body sites, so anorectal and genitourinary tracts. Uveals, the majority are from the choroidal, which is this region around here, the iris, which occurs towards the front of the eye, and then the ciliary body. So I'll start by starting at the smaller mutations, so the point mutations or substitution mutations and the small indels. So this plot shows the mutation burden in terms of mutations per megabase for the substitutions and small indels in each subtype. Each point represents a single sample, and the red line is the median for each cohort. You can see there's quite a different distribution in the various subtypes. So uveal has a very low mutation burden. The acrals and mucosals, again, have quite low mutation burdens, and this is roughly equal between them. And cutaneous melanomas have a very high mutation burden. So just mention, as I'll come back to these in a few slides, although these tumours do have lower mutation burdens, there are some outliers for the uveals and acrals, which have a higher mutation burden in comparison with the rest of their cohort. So the cutaneous melanoma has a very high mutation burden, and it is one of the most highly mutated of all cancer types. And this is just showing the mutation burden of various cancer types from a publication in 2013. So what has been known for, and this of course is this high mutation burden has been known for a long time in cutaneous melanoma, and that it is a result of mutations induced as a result of ultraviolet radiation. So this is some data from the TCGA, and again, this was known before the TCGA publication, but this is just a demonstration of it, that the ultraviolet radiation causes C to T substitutions. So this is the proportion of mutations in each context, where the red is the C to T substitutions, and you can see the majority of samples have a high proportion of samples of mutations which are in the C to T context. So as a result of, as mutations are caused by exposure to sun, you would expect the more sun-exposed sites to have higher numbers of mutations, and this is what we do see. And as you can see, this is in our cohort where we've separated the tumours by site and showing the mutations per megabase. And those more sun-exposed sites, such as the head and neck, tend to have a higher mutation burden than those which are generally less sun-exposed. So the pattern in mutations caused by ultraviolet radiation and other mutagenic processes can be investigated by looking at mutational signatures. So the first signature was developed was a single base substitution signatures. So this is where you have a mutation catalogue for the point mutations in each tumour, and you find the 5' and 3' bases for each substitution, which gives you 96 trinucleotide contexts. You then count the number of mutations in each context, and by plotting these numbers of mutations, you can see different patterns in the 96 trinucleotides, and this gives you different mutagenic processes that are causing the mutations within that cancer. So ultraviolet radiation signature was one of the first signatures developed, identified, and this was signature 7. As can be seen here, it's almost exclusively C to T mutations. But in last year, by Alexandrov, published in Nature, the UV signature was broken down into 4 subsignatures. So as you would expect, when we look at our cutaneous melanomas, the proportion of ultraviolet radiation signature in the cutaneous melanomas was dominant, and this is shown here in the red, with the grey being other signatures. And you can see that for the majority of patients, it is almost exclusively UV signature mutations, with only a small number of samples lacking any UV signature at all. Traditionally, it's been thought that UV signature is not important in the other subtypes due to the fact that they're in less sun-exposed sites. However, in recent years, we and other groups have shown that there are a subset of samples in these other subtypes which do have an ultraviolet radiation signature, and this is due to the location of where the tumour has developed. So in uveal melanoma, we see that there are 8 tumours with a greater than 50% contribution of ultraviolet radiation signature, and this is due to the fact that these are all primaries from the iris. So the iris is towards the front of the eye, and so more exposed to the sun. For mucosal melanoma, there is 17 patients with the UV signature, though it is less of a contribution in most of the tumours. And the primaries here are again from at least sites that are somewhat more exposed to the sun, so the oral and nasal sites. Acral melanomas are in 24 tumours, with about half having greater than 50% contribution. And again, it's related to site, where the most common most commonly occurs in tumours in the fingernails, but we also see it in tumours on the hand and toenails. So I mentioned previously these outlier tumours with high mutation burden, the acrals and uveals, and one of the reasons for this high mutation burden is the ultraviolet radiation signature. So this is dividing each subtype by whether or not they have a mutation UV signature and whether or not it's greater than 50%. And in the uveals and acrals, the tumours with greater than 50% contribution have a higher mutation burden. You see the similar trend in the mucosals, but it is not significant. So this explains the reason for most of these outliers in terms of the acrals and mucosals and acrals and uveals with high mutation burden. However, there still are a couple of tumours in each of these, and there are explanations for these ones as well. So in the uveal, it is both of these tumours have germline MBD4 mutations. So they have nonsense germline mutations. And these mutations have also previously been reported in uveal melanoma and in tumours that have a high mutation burden. So MBD4 is a glycosylase involved in DNA base excision repair. So loss of function results in lack of repair of mutations. For the acral outliers, this is not due to a germline mutation, but you can potentially explain it by looking at other mutational signatures. So one tumour has a mismatch repair signature, so it has a homozygous deletion of MLH1, and again, loss of a mismatch of a DNA repair defect results in accumulation of mutations. The other tumour has a rarer signature called SBS32, and it's been found in cutaneous squamous cell carcinomas, which also have a high mutation burden. And this has been proposed to be a result of prior treatment with azathioprine, which induces immunosuppressant. However, there was no known history of this in this particular patient. The other mutational signatures that we identified, most of them were signatures which are quite common throughout many tumor types, but one signature I did want to mention was SBS38. So this is an interesting signature is in that it seems to be quite melanocyte specific. We don't really see it in any other tumor types. It has an unknown etiology, but it's quite interesting because it occurs primarily in tumors without a UVR signature. So this is shown here in the black, where the red is the UV signature, and it's primarily occurring in the acrals and mucosals. So a little bit of work has been done to look at the timing of these signatures. So do these mutations as a result of these events occur early or later in the mutagenic process? And this was initially looked at by PCAWG, Pecan Cancer Analysis of Whole Genomes, where they looked at whole genome sequencing of more than 2,000 tumours from different cancer types. And they showed that this is comparing the signatures in earlier clonal mutations compared with later clonal mutations. And they showed that UV signature was higher in the early clonal stages, whereas the SBS38 was higher in the later clonal stages. And this is reflected in their overall summary of the signatures they found in the various cancer types, with SBS7 being the earliest in this comparison, and in terms of early clonal events, and SBS38 being the later clonal events. So we also see a similar pattern in our tumours with UVR signature and SBS38. So here we're seeing each line represents a patient comparing early clonal and late clonal mutations. And you can see that the blue, which represents more in the early clonal events, so you're seeing more ultraviolet radiation signaling in early clonal events. So this is a very early cause of mutations in a tumour. And whereas SBS38, which is shown here in the red, you're seeing higher proportion of this contribution in late clonal stages. So it's a somewhat later event. So now I'll move on to talk about rearrangements and copy number alterations. So rearrangements, which I may also refer to structural variants or SVs, the larger scale structural rearrangements such as insertions, deletions, duplications and inversions, as well as translocations of material between chromosomes. And here we see a number of rearrangements in each subtype, again, where each point represents a single sample and the red is the median. And here we see a different pattern when we compare it with the point mutation burden. Again, uveals have a very low burden of structural rearrangements as well as point mutations, but we see changes with the other three subtypes. Cutaneous melanomas have a somewhat mid-range of structural rearrangements, whereas acrals and mucosals have very high numbers of rearrangements. So rearrangements can result in loss of function of a gene or can be activating or increased expression through fusion genes. And in our cohort, there are about 3% of rearrangements that are potential in-frame fusion genes. And when we looked at cancer genes that are known to often cause fusions, we only saw a fairly low number. That was 14. The most of these were BRAF BRAF fusions, and they occurred in all three subtypes of Acral, Mucosal, and Cutaneous. We saw 3 RAF1 fusions, an ALK, and a ROS1. So this is just an example of one BRAF fusion where we have 2 breakpoints on chromosome 7 affecting the AGAP3 and BRAF genes. So this results in the fusion of the first 9 exons of AGAP3 with the final 9 exons of BRAF. And these exons contain the BRAF kinase domain. So when this new fusion protein is expressed, you get activation of BRAF. And most of these have been confirmed in tumors that have matching RNA sequencing that they are expressed. So here we see rearrangements across the genome where we see the chromosomes from 1 to 22 and the percentage of samples with a rearrangement breakpoint in 1 megabase regions across the genome. And here we can see that there are some similarities, but also many differences between the various subtypes. So uveal is due to the fact that it has very few rearrangements. It is very quiet across the genome. There are some similarities between acral, cutaneous, and mucosal. For example, this peak on chromosome 9p, and that's in the region of CDKN2A. There are also these peaks which are common in the acrals and mucosals, but not really present in the cutaneous tumors. And these are regions on chromosomes 5, 11, and 12. So if in a similar manner, if we look at copy number alterations across the genome, we can look at the percentage of samples altered, amplified in red and deleted in blue. And we can see, again, some similarities, but also differences. So again, the uveals are quite distinct, and they have these typical loss of chromosome 3 and gain of 8q. Cutaneous acrals and mucosals have a few chromosomes that are quite very similar, such as chromosome 6 isochromosome and loss of chromosomes 9 and 10. But there are also some differences, and these come back to these same regions with high numbers of rearrangements. So these we see these peaks of amplification on chromosomes 5, 11, and 12. And so as well as having rearrangements, these rearrangements have resulted in amplified genes, including cancer genes such as the telomere maintenance gene TERT, cell cycle pathway gene CCND1 and CDK4, and the p53 pathway gene MDM2. So the reason that acrals and mucosals have a very high number of structural rearrangements is due to the presence of complex rearrangements. So this is where you get many rearrangements occurring within a focus region of a chromosome or several chromosomes. And there are a number of different mechanisms that can result in these complex rearrangements. One of these is chromothripsis. So this is where you have an event that causes catastrophic chromosomal breakage. And this chromosome is repaired, but is repaired in a rearranged manner. So you lose chromosomal material, and you can get genes that are in the incorrect order or inverted, etc. So this is an example of a chromothripsis in one of our melanoma patients, and this one is entirely focused on chromosome 4. And if you zoom in here, you can see these high number of structural rearrangements, which is shown in the lines here, and then the oscillating copy number between 2 copy number states, which is typical of chromothripsis. So there are a number of ways that we can measure complexity or determine if a chromosome is complex or not. You can use statistical methods to determine if there is localized complexity, for example, looking to see in comparison with the rest of the chromosomes in that sample, whether there are clustered patterns and whether there are a high number of SVs, such as on this chromosome here, which has a much higher number of rearrangements compared with the rest of the tumor. Analogous to mutational signatures, you can also look at rearrangement signatures. So these were first reported in breast cancer. And instead of categories based on trinucleotide context of point mutations, these are categories based on the size of the SV, the type of the SV, such as, for example, whether a deletion, and whether the SVs are clustered or not. There are a number of rearrangement signatures, and 2 of these can be considered signatures of complexity, so RS4 and RS6. There's been several publications looking at copy number signatures, and the one I am looking at, it was first published in focusing on sarcoma, and this one is based on categories here are based on copy number size, the type of the event, that is where it's deleted or amplified, and whether it is a heterozygous loss of heterozygosity or homozygous deletion. And last year there was a tool published called QIMR, which looks at non-site specific motifs from DNA copy profiles. And again, there are a couple of measures that are of complexity, STP and CRV, as well as AMP, which is amplifications. And I just come back, as I forgot to mention, CNS5 is a complex signature, copy number signature, which is characteristic of chromothripsis and other complex rearrangements. So if we look at all these measures of complexity in our tumors, these are shown here, and clustering through clustering, where red is a higher proportion of these particular measures of complexity. And you can see that we get this clustering where we get these high number of samples with a high number of rearrangements. And if we look at the subtypes, the red and the green, which is mucosal and acral, there tends to be many more of these than the cutaneous, which are in the blue and tend to be concentrated towards this other end. At the bottom here, we see measures of whether an individual chromosome per sample is considered complex, and we've used a few different ways to determine this, such as statistical methods, methods to identify chromothripsis, as well as manual review. And again, you can see this concentration in the tumors with complex signatures, as you expect, have complex chromosomes. And it's particularly recurrent on several chromosomes. So these are 5, 11, and 12. And these are the chromosomes that I previously mentioned with the high numbers of rearrangements and copy number amplifications. So all those events are as a result of complex rearrangement events on those chromosomes. So this is just focusing in on those, seeing the same thing focused in on specific chromosomes. So we have these groups of samples with high numbers of SVs, complex localized events on chromosomes 5, 11, and 12. And this is the results from mucosal, but it's fairly similar in the acrals. So here we see the percentage of samples with a structural rearrangement and copy number, in particular amplifications. And it's concentrated on chromosome 5p. And for chromosome 12, it's towards on 12q towards the centromere. And in these regions, as I previously mentioned, we see TERT, which is amplified, CDK4 and MDM2 on chromosome 12. And for chromosome 11, this peak of rearrangements and amplifications is again on the QIMR towards the centromere. And here we see amplification of CCND1, but there are also other potentially interesting genes in here, such as the kinase PAC1 and the docking protein GAB2. So this is just some examples of tumors with complex rearrangements. And not only do they have events on those chromosomes that I already mentioned, 5, such as chromosomes 5, 11, 12, they have complex events on all 3 of these chromosomes. So chromosomes 5, 11, and 12 are complex in both of these samples. And while all 3 doesn't occur in a similar sample, you often see linking translocations between the complex events on 2 chromosomes, for example, between chromosomes 5 and 12. So indicating these are potentially arising from the same event or linked to each other. So in mucosal, you particularly see chromosome 5-12 linking translocations, so chromosome 5 with the TERT amplifications and chromosome 12 with the CDK4 and MDM2 amplifications. So it occurs in 11% of cases in mucosal melanoma, and these are predominantly oral mucosal melanomas, and they have, if you just look at the oral mucosal melanoma tumors, the ones with these linking translocations tend to have a younger age of onset. So just now I'll move on to the overall gene aberrations that are occurring within the different subtypes. I'll talk about uveal separately. As you can see from previous data, they are quite distinct from the other subtypes. So uveals, the TCGA published a paper in 2017 where they characterized whole exome sequencing of 80 uveal tumors. And they divided them into 4 molecular subsets, which I'll go on to the next page on the next slide. So we see these same 4 molecular subtypes that the TCGA identified in our cohort. So these 4 groups are divided based on the presence of chromosome 3 disomy or monosomy, and whether or not they have the presence of a chromosome 8q gain, and then presence of mutations in particular cancer drivers. So whether or not they have a BAP1 mutation, EIF1AX mutation, or a mutation in SF3B1. And other proteins which are commonly mutated are GNAQ and GNA11, and this is, tends to be across all the different molecular subtypes. So I'll talk about cutaneous, acrals, and mucosals together, as although there are many differences, there are a number of similarities. And one of the most important pathways in cutaneous melanoma is the RASMAP, aberrations in the RASMAP kinase pathway. So this is just a brief figure of the RASMAP kinase pathway. We have activation through receptor tyrosine kinase, signaling through the RAS proteins and through the RAF proteins before activation of downstream pathways. And an important negative regulator of this pathway is NF1. So back to the TCGA paper of cutaneous melanoma paper in 2015, looking at whole exome sequencing of cutaneous melanoma samples. They identified 4 molecular subtypes, which was based on mutations in genes in the RASMAP kinase pathway. So they had the BRAF hotspot, which was the V600 hotspot mutations. They also had RAS hotspot mutations, the majority of which are NRAS, but there are a small number of KRAS and HRAS. NF1 is loss of function mutations primarily, and triple wild type is that category where they do not have mutations in any of these genes mentioned previously. So if we look at those same subtypes in our tumors, we see a similar pattern as the TCGA categorizations. So here we see BRAF, so the colors here don't matter too much. I just showing here that we see BRAF mutations, primarily NRAS mutations for the RAS hotspot, and NF1 mutations, most of which are point mutations or frameshift indels, as shown by the smaller rectangles. If we compare the proportion of samples that fit within these various subtypes, comparing ours versus the TCGA, the distribution is fairly even between them. We do see slightly more NF1 mutants, and this is because as we have whole genome data, we're able to identify more structural rearrangement events in the NF1. So we also categorized our acrals and mucosals based on these molecular subtypes, and we can categorize the tumors into these subtypes, but you can see the proportions are quite different when compared with cutaneous melanoma. So this is comparing this in the percentage of samples in these various subtypes, and you can see that mucosals have relatively few BRAF mutations, and it's most common in the cutaneous. RAS mutations are fairly even between the cohorts. NF1 is quite common in all the 3 subtypes, but I just broke them down into different colors here, where the in terms of types of events, where the primarily point mutations for the and small indels in the cutaneous melanomas, whereas the acrals and mucosals have a lot more rearrangements and copy number alterations, which is expected as they have a high number of rearrangements. And cutaneous has quite a small proportion of triple wild type, whereas acral mucosal have at least 30% of tumors which fall within these categories, and this is because they have mutations in other genes which are less frequent in cutaneous melanoma. So one such gene is SPRED1. So this is a negative regulator of the RASMAP kinase pathway. It is, a couple of years ago, there was a publication in Science where they did tissue-specific CRISPR technique in zebrafish, and they found that SPRED1 functions as a tumor suppressor in mucosal melanoma. If we look at SPRED1 mutations in the various subtypes, where blue is a single hit and red is double hit, and that is both alleles affected. We see that the acrals and mucosals have a high percentage of samples with spread 1 mutations, in particular the double hits. So KIT is another gene mutated, and it is a receptor tyrosine kinase, and it can activate the RAS-MAP kinase pathway. So this is below is showing activating missense mutations and in-frame indels in the various subtypes. We can see that cutaneous melanomas have fairly low frequency, but they're more common in acrals and particularly mucosals. Another pathway which can be important in these subtypes is the cell cycle pathway. So these genes involved in progression of cells through the cell cycle. Here I'll just focus on CDKN2A, CCND1 and CDK4. So CDKN2A aberrations are quite common in all 3 subtypes, and they're about the same proportion in each cohort. And I just mentioned here, I previously mentioned we have methylation data. We'd look to see if there was any promoter methylation in any of the loss of function genes, as promoter methylation can result in decreased expression. For most of the tumours that we had methylation data for, we didn't really see too much associated, but with CDKN2A, we did see 12 tumours with promoter methylation and therefore decreased expression. So if we come back to CCND1 and CDK4, so I've already previously mentioned these genes are amplified as a result of complex rearrangements on chromosomes 11 and 12, and as is reflective of the fact that cutaneous melanomas don't have as many rearrangements, we can see a smaller percentage of samples with amplifications in this region. And it's very frequent in both the acrals and mucosals, with acrals tending to have more of the CCND1 amplifications and the mucosals having more of the CDK4 amplifications. Another pathway that is important in many cancer types is telomere maintenance. So cancer samples are often associated with longer or shorter telomeres when compared with normal tissue from the same person. An important stage in maintaining cancer immortality is maintaining telomeres. And there's a number of ways to do this. The most common is the reactivation of telomerase pathway, and you often see this in the form of TERT promoter mutations. There's also an alternate lengthening of telomeres pathway, and it seems to be connected to loss of function mutations in the genes ATRX and DAX. So we see a small number of loss of function mutations in ATRX, but we do see them, and these are primarily in mucosal melanoma tumours. And we identified this as a significantly mutated gene when we published our paper last year. For TERT aberrations, there are a number of different mechanisms of TERT reactivation. So there's the promoters, amplifications, and rearrangements. So for TERT promoter mutations, there are specific sites which are commonly mutated. And if we look at in comparison, although these promoter mutations do occur in the acrals and mucosals, they're much more common in the cutaneous melanoma, again reflective of their higher mutation burden in terms of point mutations. So TERT amplification, that is copy number gain, is also another way of gaining TERT reactivation. And I previously mentioned TERT in the context of chromosome 5 complex rearrangements. So again, we see more of these on the acral and mucosal, in the acral and mucosal subtypes than we do in the cutaneous, due to the presence of these complex rearrangements on these chromosomes. Finally, we have TERT rearrangements. So as a result of the rearrangement, you can have a relocation of a super enhancer element in close proximity to TERT when you have an upstream event, an event that's upstream of TERT. And this can result in increased expression of TERT. And this seems to be a feature in acral melanoma, so TERT enhancer hijacking. So this is showing the acral tumours with RNA sequencing, and this is the different types of TERT aberrations, where the blue ones are events where there's an upstream rearrangement upstream of TERT, and the darker blue is those which are on rearrangement plus amplification, and this seems to be associated with higher TERT expression. So if we look at all these events together, we can see that TERT is a common aberration in all three subtypes, but the type of events are different, with the cutaneous being primarily promoter, acrals and mucosals having amplifications, acrals having the upstream SVs, and the black represents when there are multiple TERT aberrations in the same tumour. So just briefly mentioning a couple of other genes that are more focused on one subtype. So this is SF3B1 in reference to mucosal, and these events are all occurring within the same codon. And SF3B1 is a component of the spliceosome, which plays a key role in the early stages of pre-mRNA splicing. In mucosal melanoma, it primarily occurs in lower body site tumours. So I'd already mentioned SF3B1 in the context of uveal melanoma, but it's rare in cutaneous melanoma and absent in acral. So, and another gene which is particularly focused in acral melanoma is TYRP1, and this is were all frameshift mutations, and it's quite interesting because 6 of the 7 were a single frameshift variant. TYRP1 is an enzyme involved in the generation of eumelanin in melanocytes. It's also pathogenic in ClinVar for oculocutaneous albinism type 3, which is autosomal recessive disorder of melanin biosynthesis, and it reduces pigmentation of the hair, skin, and eyes. In recent years, it's been reported to have a non-coding function and regulating gene expression by activating as a sponge for miRNAs, including MIR16. But exactly what its role in acral melanoma, if any, remains to be determined. So just finish off by looking at treatment options. So this is not an exhaustive list. This is just touching on a few different treatment options for the rarer subtypes, as there are fewer treatment options available for these subtypes. And this is just highlighting some of the ways that genomic data can highlight potential treatment options for subsets of samples based on our cohort. So BRAF inhibitors have been used in cutaneous melanoma for some time, and often in conjunction with MEK or in the presence of also MEK inhibitors. And in our cohort, we see the BRAF V600E mutations in cutaneous melanoma has been well established. We also see it particularly in smaller rates, particularly in acral melanomas. So these can also be targeted. And so BRAF inhibition has been reported to be effective in metastatic melanoma, regardless of subtype, in a Korean study. So immunotherapy by immune checkpoint blockade has had a profound impact on the survival of cutaneous melanomas. And this is just showing results from a clinical trial a few years ago where they had shown progression-free survival in the presence of anti-CTLA4 or anti-PD-1 antibodies, or with a combination, resulting in improved progression-free survival. And this is just showing some immunotherapy results from our cohort. We have a subset of patients who have later undergone immunotherapy after their sample has been collected. So there are 77 cutaneous tumours. And if you divide these into good and poor responders, you see this expected better progression-free survival. And genomic data, a lot of it work in the last few years has been gone into looking for molecular markers of response to immunotherapy. And one that has been identified is tumour mutation burden, where if you have a higher mutation burden, you tend to have a better response to immunotherapy. And this can be seen in our cutaneous melanoma patients as well, where the good responders have a higher burden than the poor responders. So immune checkpoint in other subtypes has not been as well studied. And from this data, we showed that the other subtypes, there are subsets which have higher tumour burden. And so could these tumours be potentially responsive to immunotherapy? So this is these outlier tumours, particularly in the acrals and uveals, that have higher burdens due to the result of ultraviolet radiation signature, MBD4 mutations, and other signatures such as mismatch repair. And so potentially with a higher tumour burden, they could respond to immunotherapy. Unfortunately, we don't have any response data from these patients in this cohort. Prolonged stable disease has been reported previously in a uveal patient with an MBD4 mutation, however. Another potential treatment is with CDK4 and 6 inhibitors. So these are used in the treatment of some breast cancers, and these inhibitors target aberrations in the mostly CDK4 amplifications and CCND1 amplifications. And Acrals and Mucosals have a high percentage of samples with these amplifications, so they could potentially be responsive to CDK4 inhibitors. And there was a report a couple of years ago with preliminary activity in acral melanoma in patients which had CDK4 aberrations in their tumors. There's also been a couple of papers from groups in China looking at effects of these inhibitors on PDX models. And this is just showing the effect on tumor volume in papers from China in mucosal and acral. And the tumor volume is decreased in the mucosals and the acrals in the presence of inhibitor. So just to summarize the overall results of this study, if we look at mutation burden, we see differences between the various subtypes. So uveals have extremely low point mutation and rearrangement burdens, whereas cutaneous has a very high mutation burden, but only a somewhat mid-range of rearrangements. The acrals and mucosals have low mutation burden, but a high number of rearrangements. UVR signature is dominantly in the cutaneous melanomas, but is also present in subsets of samples based on the primary site of the tumor in the other subtypes. Complex rearrangements are rare and less common in the cutaneous and uveal cohort subtypes, but they're common in the acrals and mucosals, particularly focusing on chromosomes 5, 11, and 12. For the TCGA subtypes, BRAF is most common for the cutaneous melanomas, and the acrals and mucosals have more of the triple wild type subtypes in comparison with the cutaneous. TERT aberrations are common in all three subtypes, but the types of events, the way that these aberrations occur is different in the different subtypes. Uveals are quite genomically distinct from the other cohorts, with loss of chromosome 3, 8q gain, and mutations in only a small number of cancer genes such as BAP1, GNAQ11, and SF3B1. For acrals and mucosals, KIT mutations and SPRED1 mutations are more common, as are CDK4 and CCND1 amplifications. Acral has TYRP1 mutations, and mucosal has SF3B1 mutations. In terms of treatment options for the rarer subtypes, there are subsets which could be targeted, the BRAF V600 mutant tumors for immunotherapy, those high tumor burden tumors, and for CDK4 and 6 inhibitors, the CDK4 and CCND1 amplified tumors. So just to acknowledge everyone involved in this work as a very large-scale project that's been running for many years. So I'm based in the Medical Genomics Lab, which is led by Nick Waddell in QIMR Berghofer, and Nick Hayward in the Oncogenomics group, and Peter Johansson are also one of the leaders of the AMGP in conjunction with Graham Mann and Richard Scollier. And James Wilmot is also heavily involved in the AMGP. For the clinical data, a large number of clinicians have contributed. As I mentioned, we have collaborators in the University of Colorado, Switzerland, China, Sweden, and for mucosals, which I didn't talk about here, but we also had exome data at validation cohort from University of California. And finally, thank you all the patients and their families.
Okay, so we can do questions in the chat, and then if we're done with that, people can feel free to ask questions out loud. So the first question we have in the chat, Felicity, is one, what was the minimum depth to reliability call to call a variant, and are there additional criteria that variant being added to the counts of mutational signatures?
Yes, so we have a pipeline that uses 2 variant callers, and each of those have their minimum coverage. I can't remember exactly, but we have a minimum coverage. I think it's 10 or 20 reads in the tumor and 10 or 20 in the in the normal, and we need adequate coverage in both the tumor and the normal to be confident that it's a somatic mutation. So after we've done our calling, we do additional filtering for not only coverage, but also other features to remove variants. And in terms of mutational signatures, we also exclude any of the common variants, so the really based on population frequencies and things like this, which may be artifacts that have escaped through the process.
Great. And then the second question from Edward Evans, considering that whole genome sequencing is being used, does this bias mutational signature counts to mutations that occur throughout the entire genome while potentially hiding more localized and potentially more deleterious signatures? Is there any filtering for genome-wide mutations or normalization for this?
So we don't do any specific genome-wide filtering for those things. So yes, it could potentially hide signatures, and in particular, you can get transcription. The subsignatures have a transcription bias. So if you look on the forward strand and the reverse strand, it tends to be more dominant in one and the other, and UV signature is actually one of those. So if you do focus in on just exome-based events, you can see this transcriptional, and you may miss that in the whole genome. But when we've done quite a lot of work comparing whole genome and exome, and even from the same sample, and we haven't really seen any difference in terms of what we see. The downside with exome is that you have a smaller number of mutations, and once it becomes a smaller number of mutations, it's harder to identify signatures, particularly if some of the signatures are quite dominant, like UV signatures are fairly easy signature to identify as it's quite strong, whereas, for example, the BRCA signature, which is signature 3, is very weak across many nucleotide contexts. So when you have the smaller number of mutations, it can be harder to identify those ones correctly. But yes, in terms of comparison, we haven't really seen any differences for when we have looked between them.
Okay, so the next question is from Tom Braun. Two general questions about building the data set. One, what are the proportion of melanomas make it to the labs? Are the providers obligated to send in tissue samples? Is there data regarding initial, DX prognosis maybe versus metastatic disease, particularly those who respond to immune checkpoint inhibition and then later progress?
So in terms of proportion of melanomas that make it to the labs, I'm not 100% sure in terms of proportions, but we're very lucky in that the majority of our samples have come from the Melanoma Institute of Australia, which is in Sydney. And so this has been specifically set up to accumulate a tissue bank of tumours. And so I don't think anyone's obliged to send them in, but they have agreements with pathology centres with informed consent of patients to send in these samples. So they have a tissue bank. Not everything makes it to sequencing because of, in terms of whether there's enough DNA to extract. But again, we're also very lucky in that they have a very good clinical database. So not all of our samples have that because we have them from different sources, but those ones are very well clinically annotated, and they have done a lot of work, not just necessarily with sequencing, but with melanoma in general. So there is some diagnosis data with regard to initial diagnosis versus metastatic disease for those ones. And certainly there are questions that we can ask about that, and that we have done, and that certainly Melanoma Institute of Australia has also done.
And then so from Christopher, great presentation. He asked, did you observe any melanomas with the translocation in the Q12 chromosome 12 region of the Y chromosome to the P arm of chromosome 22? And where would we find a list of these germline mutations that you found in these different types of melanoma?
So we haven't specifically looked. I hadn't noticed it as one that was particularly recurrent, but I also couldn't say 100%, but because I haven't looked, I'll certainly have a look now to see if we do. But there's certainly lots of SVs, so there may be one or two stray ones, but it's certainly not one that immediately leapt out, but doesn't mean it's there, not there. And in terms of list of germline mutations, so in terms of the full germline list, that's not published yet. We're working on more germline data at the moment. The raw data is available for all of our published ones, so you can download it and do the germlines yourself. But in terms of, because it's germline data, it's also in terms of ethics, you usually generally need to get more, you need to show your ethics approvals in order to show that. In terms of a short list, we're working on doing some more germline stuff now.
Great. So our next question is from Casey. Do you see any specific genomic features of uveal melanoma that might explain the particularly low immunotherapy response in this subtype?
Well, the very low mutation burden is, they're extremely low. They're not just low compared with the other types of melanoma. They're one of the lowest of any cancer type. So that's one reason. I don't know about in terms of PD-1 expression, but that's the usual clinical marker for looking at uveal. And I haven't personally read about that myself, but probably I would assume that they would also have a low expression of PD-1 that might also explain the low immunotherapy response for those tumours.
And I just had one question kind of stemmed off of that, because you did show that in cutaneous, your higher tumors with the tumor mutational burden responded better to immunotherapy. Since you have all this great clinical data, is there any other genomic features other than tumor mutational burden that corresponded with immunotherapy response that you guys have seen?
Yeah, so we are actually working on a paper at the moment that's been submitted and is under review. And so we didn't really see anything that was brand new and this is completely novel. We sort of seen what other people have seen. So as I mentioned, PD-1 expression. So we also had immunohistochemistry as an RNA sequencing for those 50 of those 77 that I mentioned. And so we see this increased PD-1 expression in the good responders, which again is quite well known. It's used clinically to identify the responders. And in the RNA sequencing, interferon gamma related signatures. So those have been reported in a number of other subtypes as well. And that's with the most best predictors in ours was the interferon gamma and tumor burden, which is not new, but it's also been shown by other people. But yes, we see what other people have seen as well.
Then we had
PD-L1. Yes, sorry, PD-L1 expression, yes.
Great. Well, if anybody else has any other questions, feel free to ask or type them in the chat. And just a reminder to students and postdocs who would like to stay on with a meet and greet with Dr. Newell, you can just stay on this Zoom and we'll start that around 1 o'clock. Thank you so much, Dr. Newell. That was really excellent data.
Thank you for inviting me.